Good morning, everyone. Today we are going to discuss the translation. So, in this module, we are going to discuss about the translation. So, what do you mean by translation? Translation is the process of assembling of amino acids as per the codonic sequences in mRNA within the ribosomes, that is in the cytoplasm, or we can say it is a synthesis of protein from based on the amino acid assembly. So here it refers to the process of polymerization of amino acids. So how the amino acids are assembled or connected with each other. It is with the help of peptide bond. So peptide bond, you know, the amino acid terminal, carboxyl terminal of one amino acid with the amino terminal of another amino acid with the help of CONH bond. So that CONH bond is called as peptide bond. So as many amino acids will be assembled one after another, and in between the carboxyl terminal and the amino terminal, the peptide bond or CONH bond will be produced, will be formed, and that process of polymerization of many amino acids according to the codonic sequences what is given, what is in the mRNA from the direction of 5 to 3 direction, and that entire process is collectively called as the protein synthesis, and the chain of amino acids is formed as a result of polypeptide formation, that is collect, we'll call it as a single polypeptide. So here the location, when we mention about the location, it is in the ribosomes within the cytoplasm. So the order and the sequence of amino acids are defined by the sequence of bases, sequence of codons, we can say triplet base sequences, codonic sequences within mRNA. So we have learned already the characteristics of genetic code. So they are continuous and it is read in a triplet manner. So each triplet of bases from 5 to 3, in that order, triplet base sequences, the amino acids will be assembled with the help of tRNA. So that process is collectively called as translation. The amino acids are joined by a bond which is known as peptide bond, and the formation of peptide bond requires ATP. So all these polymerization requires the presence of ATP. So here the next slide, when we are saying the steps, the major steps, when we say the first and foremost step is because within the cytoplasm amino acids are in an inert state, it will be floating. So for activating the amino acid as well as for binding that activated amino acid to the respective tRNA, that step we'll call it as charging of tRNA, or another one, preparatory step. It is a preparatory step before the initiation of the translation. So that step is called as charging of tRNA, or we can say amino acylation of tRNA. So this amino acids will be moved or will be floating within the cytoplasm in an inert way. So for that, for activating the amino acids, then only if that in the presence of energy, that means ATP. So first the amino acids will bind with the ATP in the presence of a specific amino acid synthetase enzyme. So each and every amino acid, there are 20 amino acid types of amino acids are floating within that one, and each amino acid will have its own respective enzyme within the cytoplasm. So that amino acid will bind with the respective amino enzyme in the presence of ATP. So that step is called as activation of amino acid. Then this activated amino acid, so they will form an amino acid enzyme AMP complex. Amino acid enzyme AMP complex. That amino acid enzyme AMP complex will bind with the respective tRNA. So what do you mean by the respective or cognate tRNA? In the textbook it is written. The meaning of cognate tRNA means there are 20 amino acids, and in eukaryotes maximum of 33 tRNAs. And in prokaryotes means 22 tRNAs. That means each and every amino acid will have its own respective tRNA. So here AUG, that is methionine, is the amino acid, means the tRNA, corresponding tRNA in the anticodon site, they will have UAC. Okay, UAC. So that binding, that corresponding binding of amino acid with activated amino acid on the 3 terminal of tRNA. in the presence of that amino acid enzyme, AMP complex will bind with the third end of the tRNA, which carry the corresponding anticodon. So if it is methionine to bind on the third end, means the anticodon will be UAC. If it is phenylalanine is bound to the third end, means that the anticodon will be AAA, because their codon is U. And if it is glycine, means anticodon will be CCC, because GGG stands for glycine. So anticodon, so this anticodon and mRNA, there will be a collinearity. So that process is called as charging of tRNA. So here I will read the charging or aminoacylation of tRNA, not rRNA. tRNA is the amino acids are activated in the presence of ATP and linked to specific tRNA. Then soon after the activation of amino acid or aminoacylation of an amino acid, that means the tRNA will carry the corresponding amino acid at the third end. Then the next step is, first step of that translation, when we are saying it is the initiation. So what do you mean by initiation? Means here we will move on to this slide. So here this slide represents the activation of amino acid. So already I said the corresponding amino acid in the presence of ATP and in the presence of corresponding enzyme, that only E, and that all these binding takes place or mediated in the presence of magnesium. So in the presence of magnesium, amino acid AMP enzyme complex will be formed by releasing inorganic pyrophosphate. Then this activated amino acid will bind with its respective tRNA, specific tRNA, and then they forms amino acid tRNA, or we can say charged tRNA. So in the figure also, this it is very clearly mentioned. So here it is enzyme and here it is ATP. So here ATP and then the enzyme complex, enzyme and amino acid will bind and they will form an enzyme amino acid complex. Then this enzyme amino acid complex will bind with the tRNA and the tRNA will be charged with that corresponding amino acid, or we can say charged tRNA. Then the next slide when we are moving, that is the initiation. So in the initiation step, first the mRNA will be lying in the ribosomes. It will bind with the ribosomes. So here the ribosomes, normally it will be found floating within the cytoplasm in the form of a dissociated form. So small subunit will be and large subunit will be in a dissociated form. They will be separated state only. So soon after the RNA reaches the cytoplasm, with the 5 end of the RNA, it will bind with the small subunit. Here in the figure you can see the binding of small subunit with the mRNA at the 5 terminal. And then the charged tRNA, specific of initiation codon. Specific of initiation codon means always initiation codon is AUG. That means the tRNA which carry the methionine with anticodon UAC will bind, and then that forms a site. That site is called as P site. So P site is always the site within the ribosomes which allows the binding of first tRNA, and thereafter in that particular site only the peptide bond formation takes place. Hence that site is called as P site. So immediately after that binding of tRNA with the 40S ribosomes, then the larger subunit also, corresponding larger subunit also binds with, and there forms the complete initiation complex. Then the larger subunit, that means 60S subunit also, ribosome also now combines with the 40S ribosomes complex in the presence of magnesium. So that step is called as, all these process takes place in the presence of ATP. ATP is required for the initiation. Even for the activation of tRNA, at the same time initiation step also ATP is required. Then the next step, so as soon as the first methionine or the first codon, first amino acid, that is methionine, when with the help of tRNA, it will bind to the codon of the mRNA. So there will be a codon-anticodon base pairing takes place. So as a result, the first amino acid will be assembled in the P site. Then the elongation, we can say. In the elongation step, by the time the second tRNA will be assembled, the next codon. Second tRNA will be assembled in the next codon of the mRNA. So if it is UUU, so then automatically the tRNA which carry AAA and at the third end of the tRNA, which will be phenylalanine. They will come and assemble next to the P site. So that site is, second site is called as A site, acceptor site. Why it is called as acceptor site means always the new tRNAs will come and along with the amino acid will come and assemble at the A site. So there are two sites will be formed. One site is called as P site and another site is called as A site. P site is for the formation of peptide bond as well as the assembly of the first tRNA with the first amino acid. And A site is for accepting the amino acid with the tRNA and there the anticodon base pairing will take place. So when the second amino acid along with the second tRNA also is charged with assemble at the A site, there the P site and the A site there will be one amino acid. So the amino acids in the P site and the A site, there the peptide bond formation will take place. So the peptide bond formation is taking place in the elongation step. So the peptide bond formation between the methionine and the second amino acid, say for example, if it is phenylalanine means between the phenylalanine with the help of enzyme. So there the mediation of the enzyme is peptidyl transferase. Then again, the ribosomes moves over. So as soon as the dipeptide formation is taking place, the first tRNA and the first amino acid, the bond will break with the help of energy. So that bond will break and release the energy. Okay, that bond, the first tRNA and the first amino acid. So that means the tRNA 1 and the methionine. There the bond will break, and then it will the uncharged tRNA will move out of the site of P site, and then ribosome along with the second tRNA and the dipeptide at the third end will move one codon ahead in the three direction. So that process of movement of ribosomes one codon ahead towards the third end, that process is called as translocation. So here the dipeptide formation takes place and the detachment of the first amino acid with the first tRNA, and the tRNA will be moved out of the site into E site. That site is called as exit site. So exit site, from there again it will move out of this. Meanwhile, the ribosome will move one codon ahead. So the second tRNA along with the two amino acids or dipeptide chain will move from A site to P site. So the A site will be free by the time the third amino acid along with the third tRNA will assemble at the A site. So in this way, continuously the peptide bond formation takes place by the movement of the ribosomes towards the third direction, third end of the mRNA, one codon ahead, it will be proceeding. That step is called as elongation. So here in the figure I have given, that is here the first tRNA and initiation step, that means the first tRNA at the P site and the second tRNA at the A site, and both will carry one amino acid. So first amino acid will be always methionine, second amino acid accordingly. So the tRNA with the first between, since these two amino acids are lying side by side, there is a formation of dipeptide bond between that. As soon as the dipeptide bond is formed between the tRNA, first tRNA with the first amino acid, that bond will break and it will move out of the site. Simultaneously, the ribosome will move ahead towards the next codon, one codon ahead towards this one. So there will be a translocation. That means the second tRNA with the dipeptide will shift from A site to P site. So the A site will be left free. The third amino acid carrying tRNA will assemble. There the codon anticodon base pairing takes place, and like that one after another. Here you can see how the assembling of amino acid is taking place. This process will continue, and then as a result elongation will take place. So here the next one again comes, the second tRNA will detach, the tripeptide bond formation will take place. Third tRNA also, second tRNA also move out of the site to E site, then to out of this one. The fourth codon will assemble at the A site, then the fourth tRNA with the fourth amino acid will assemble. In this procedure, it will be continued. So here you can see the ribosome proceeds to the elongation phase of protein synthesis. During this stage, complex is composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. That means codon-anticodon base pairing. The ribosomes moves from codon to codon along the mRNA towards the third direction. Then amino acids are added one by one, translated into polypeptide sequences, and which is dictated or which is supervised by the DNA. How it is supervised by DNA means DNA only producing the mRNA. So the mRNA, the codonic sequences. What type of codonic sequences are there in the mRNA? Accordingly, the type of amino acids also assembled. So here we can see the messages carried from DNA, which is located within the nucleus, and as per the instructions, it will be coded in the form of or transcribed in the form of mRNA, in the form of triplet base sequences, codons we can see. As per the codonic order, the tRNA will assemble the respective amino acids into the ribosomes. So the ribosomes helps in assembling and as well as the formation of peptide bond between the amino acids. So the whole process will elongate or will be continued or will be carried out in the form in towards from 5 to 3. 5 dash to 3 dash direction, or 5 prime end to the 3 prime end. Now we'll move on how the termination is taking place. So as the assembling of amino acids proceeds towards the third end, and at a particular point where there are stop codons. So there are 3 stop codons we can say, that is UAA, UAG, UGA. So when the A site, when the ribosome reaches the A site where the codonic sequences are UAA or UAG or UGA, any of these stop codons, there is no corresponding tRNA for the stop codon. So that will give the indication of which one, indication of termination. Since there is no tRNA to bring or to assemble the amino acids in the presence of termination factors. Here, initiation also there are certain factors are needed. Initiation factors, elongation factors, same simultaneously. For the termination process, termination factors. So the termination factors will assemble, and there is no corresponding tRNAs because they are exposed to the UAA or UAG or UGA. So at the end, the releasing factors, or we can say termination factors, releasing factors, will bind to the stop codon, and it will terminate the translation and release the complete newly synthesized polypeptide out of this. It will be peeled out by the breaking of the amino acid bond between the amino acid and the last tRNA. So that bond also will break, and then the dissociation of ribosomes as well as the peeling out of mRNA from the translation site. So the synthesized polypeptide is released with the help of releasing factors. So the whole entire process will come to an end. So here, this is the diagrammatic sketch. In the first one, it is initiation. Once again, a recap. The initiation means the binding of 40S subunit of ribosome onto the 5' end of the mRNA. Then the first tRNA, which carries the first amino acid and assemble at the first codon, that means AUG. Simultaneously, the largest subunit also will bind in the presence of energy and initiation factors. So they will form an initiation complex. And as soon as the initiation started, then the next step we'll call it as elongation. So the first tRNA with the first amino acid is assembled to the first codon, and that site is called as P site. The second tRNA, which carry the second amino acid, will be assembled at the second codon of the mRNA, which is located within the ribosome. And that site is called as A site or acceptor site. Since 2 amino acids are lying side by side between the amino acid, the peptide bond formation is taking place. Soon after the dipeptide formation, the bond between the first tRNA and the first amino acid will break, and the uncharged tRNA will move out of the site. Simultaneously, the ribosomes roll over one codon ahead. So the second tRNA along with the dipeptide will move to the P site and the A site will be free. The third amino acid carrying tRNA will assemble. Again, tripeptide peptide bond formation will take place. The second one will be moved out of the site. The third one will be shifted to the P site. The fourth one will assemble at the A site. So it is mediated with the help of initiation factors, and the whole procedure will continue till so that process of transfer of or the translocation of ribosome one codon ahead along with the uh, tRNA and the elongated polypeptide. That process is called as translocation, mediated with the help of an enzyme. That enzyme is called as translocase, whereas peptidide transferase enzyme mediates the peptide formation. Then when it reaches the stop codon, the site reaches the stop codon, UAA, UAG or UGA. There is no corresponding tRNA. So here the releasing factors will bind, and that will result in less affinity of mRNA to the tRNA. There is no corresponding tRNA, so the dissociation of ribosomes will take place. The newly synthesized polypeptide will detach from the last tRNA, and then that last tRNA will be removed and will be moved out of the site, and the growing polypeptide also, or the completed polypeptide, will be peeled out of the ribosomes, and the dissociation of ribosomes also will take place. So in that way the entire process will come to an end. So this is the gradual changes or pictorial diagram which shows. So here in this first one, it is the initiation complex formation. So that is here the small subunit, here the large subunit, assembling of first charged tRNA. That means methionine carrying tRNA will assemble at the P site. Then the A site also will be there. To the A site, the second tRNA will assemble. And in the third diagram, you see the amino acids, the detachment of the peptide bond formation between the first amino acid and the second amino acid with the help of peptidyl transferase. And not only that one, immediately after the dipeptide formation, the first tRNA and the first amino acid, the bond will break. That energy is utilized for translocating the ribosome one codon ahead. So that process is called as translocation. And here you see the dipeptide. So the tRNA, second tRNA from A site will be shifted to P site. Here, A site to P site. So in the P site, there will be dipeptide with the second tRNA. First tRNA will be moved out of the translation site. Then the third tRNA with the third amino acid also will come and assemble at the A site. Then again, the entire process of peptide formation, sequential formation of peptide bond formation will continue. And the process will continue till it reaches the stop codon. So here you can see the stop codon UAA. So there is no corresponding tRNA. So here in this tRNA, the growing polypeptide will be there, last tRNA, but there is no corresponding tRNA. So instead of that, releasing factor will bind. In the presence of releasing factor, the last tRNA with the growing polypeptide chain or with the polypeptide chain, that bond will break, and that will result in dissociation of subunits and the detachment of last tRNA from the mRNA, as well as the growing polypeptide also left free out of the site, and releasing factor also will be moved out of the site. So the entire process will come to an end. So then accordingly, these are the main steps I wish to summarize in this step. So the first step is activation of amino acid. Amino acid in the presence of ATP, in the presence of enzyme and magnesium. Amino acid AMP enzyme complex will be formed. That amino acid AMP enzyme complex binds with tRNA, cognate tRNA, which carry the first amino acid. And they will form the amino acid tRNA complex. Plus enzyme will be removed, as well as the AMP also will be removed. Then initiation. Small subunit of 40S ribosome binds with the mRNA. Charged tRNA specific for initiation codon reaches the P site. Larger subunit of ribosome, now combines with 40S mRNA and tRNA methionine, because always the first tRNA will be carrying methionine because the codon is AUG. So complex in the presence of magnesium. Then in the elongation step, the second tRNA charged with amino acid, second amino acid occupies the A site. Then peptide bond formation between the methionine and the second amino acid with the help of enzyme peptidyl transferase. Then ribosomes moves over mRNA in 5'3" direction and the process will continue. And at the last step, the termination, there the releasing factor help in dissociating the ribosomes as well as the last tRNA with the polypeptide chain, and then the process will come to an end. And once this protein polypeptide is formed, then it can be folded because it is only a primitive form or maybe in the primary structure. Then according to the cell's need, it can be converted into secondary structure, that is either in the alpha helical form or in the beta pleated form, then into tertiary structure with the help of the bond between the same polypeptide chain, and further it can be folded into quaternary structure. All these folding of proteins or processing of polypeptide into proteins is done with the help of another type of proteins that is called as chaperones. So it can be either utilized as structural protein. So we have learned in last year structural proteins, say for example actin and myosin, or it can be used as functional protein, that is antibodies, protective in function, or it can be even used as hormones, that is insulin. Insulin is a typical example proteinaceous hormone. So according to the cell's need and the function, that particular protein or polypeptide will be folded with the help of a special folding proteins that is called as chaperones. Then that will be processed into fully functional form and then released into the cytoplasm, and from there into the Golgi bodies for packaging and folding, and from there into through the endoplasmic reticulum into the respective sites where it is required. So the entire process of formation of protein as per the codonic sequences in the mRNA is called as translation. So with this, we'll come to a conclusion. Thank you very much.